Welcome back, Biology 220. Today we're going to be talking about bacterial, normal flora, and respiratory diseases. So that word flora, we think flora and fauna, we think the plants and animals that make up an environment. So we're talking about bacterial flora. We're talking about the normal bacteria that make up our bodies that are in different regions of our body. And we'll also talk about some respiratory diseases. So, not all bacteria are bad. Actually, most are not bad. Only a small subset of bacteria actually cause infections and can cause issues with our bodies. So here's a list of just normal bacteria that you would have by body region, right? We think in the intestines, we think about E. coli, right? You can get food poisoning from E. coli, or you can get it from water. I used to test water, and we would test for E. coli in streams that were near areas where cattle would graze in Raglan. And when it would rain and we'd test the water, there would be a lot of E. coli growing in the water to the point that it was unsafe to swim in because it could cause ear infections, it could give you pink eye, or if you got some of it, you could breathe it in, or you could get food poisoning from it. But in your intestines, it's actually healthy and normal to have that bacteria in your intestines. So some of these bacteria, where they naturally are, is normal, but in other parts of your body, it might cause infection if that area isn't going to be conducive for that bacteria. So normal flora, that would just be these organisms that normally inhabit these parts of our body, and they do not cause harm in those areas. So what's amazing about this is they used to think this was different. They used to say we had 10 times as many microbes, bacteria, in our body than we have actually human cells. Recently, they did a study here in the bottom. I have a little picture of that, but they think now it's actually closer to a ratio of 1-to-1 with slightly more bacteria, but close to a 1-to-1 ratio. So one estimate was that we had 30 trillion human cells and about 39 trillion bacterial cells. But either way, even with the new studies, they say we have more bacterial cells than human cells, which is pretty amazing. And these microbes are found all over our bodies. Any part of your body that is exposed to the environment or connected to the environment is going to have some amount of bacteria in there for the most part. And we'll look at some exceptions. So some areas where we're going to have normal flora of bacteria and microbes would be the skin. You'll notice the name of this one, Staphylococcus epidermidis. We think of epidermis because this is where these organisms live. So superficially on the skin, you have organisms in your digestive tract. Pretty incredible. It says up to 40% of the fecal mass is microbial cell material. And that's a little bit misleading. So fecal matter is going to be about 75% water and 25% is going to be solid. So that 40% is talking about the 25% that is solid mass minus the water. But still, that's really significant that 40% of that dry fecal mass is purely from these microbial cells that have died or are in the fecal matter. So respiratory tract, right, that's connected to the environment. We're going to have normal flora in here that you see here listed. And then genitourinary tract, you're also going to have some normal flora. So here, it's talking about the female urethra should be between 3.5 and 4.5 on the pH scale. And you'll notice a lot of different areas of our bodies, there's certain pH and salinity values that prevent certain bacteria from growing, and there's certain nutrients that allow particular bacteria to grow. And as long as those conditions are normal and they're in balance, you typically do not have pathogenic bacteria living in those areas. But if something happens that gets your body out of those ideal ranges, then you become more prone to infection. So some areas of your body that should not have bacteria, or at least a very small amount, would be your lower respiratory tract. So your lungs, bronchi, trachea, your internal reproductive organs. You shouldn't have bacteria growing in your testes and ovaries. Your upper urinary structures, so bladder, ureters, and kidneys, should be free of microbes. And then your blood, of course. And then, of course, a healthy developing fetus should not have microbes in it. And there's various reasons for this. And all the conditions in these areas prevent bacteria from being able to normally grow. Right? In the urinary tract, you have an acidic substances pushing through those, and it's always an outflow. So that prevents bacteria from growing. In your blood, there's not much free iron that bacteria need to grow. It's bound up in the hemoglobin. So all of these different environments, nutrients, are going to prevent certain bacteria from growing. So we have these two groups for our normal flora. We have what we call a resident and what we have that we call transient. So just like a resident at a university would live there over a long period of time, a resident microbe would be one that is almost always there. For example, like we talked about E. coli in your intestines. Normally, it's always going to be there. Transient normal flora would not be those. They don't cause disease, but they're not always there. So some you might pick up from particular foods and they pass through. They might stay around for a little while, but they're non-pathogenic. It's normal. They're just not necessarily always going to be present. So for the bacteria in our bodies, they're constantly battling each other. There's competition. And we call this microbial antagonism. Just like the antagonist in a story would be, working against the protagonist, antagonism is when things work against each other. So the normal flora inhibits pathogens by taking up space, using up nutrients, and changing the pH in those particular parts of our body. So we call this good bacteria. You've probably heard that before. So if the good bacteria is there, then there's nowhere for the bad bacteria to live. So the good bacteria covers all the structures, and if the bad bacteria gets in there, it has nowhere to attach to our bodies. And the good bacteria is using all the nutrients, and it acts as a barrier between our bodies and the bad bacteria. So that's why you'll hear people, after they've taken antibiotics will kill good bacteria and bad bacteria. So people will take probiotics to try to restore that good bacteria to prevent the bad bacteria from infecting them again. So we have this balance between the normal flora and these pathogens, because some of these pathogens will be there, but they're in such small amounts that they're in balance with the normal flora of our bodies that they do not cause disease. And they might be an extremely low, population size. But then you take antibiotics, it screws everything up, and now the bad bacteria have an opportunity to come in, grow, and infect our bodies. So one example of this would be the presence of normal microbiota in the adult female vagina maintains a local pH of 3.5 to 4.5. But if something happens, like we mentioned with antibiotics, then it could cause a yeast infection, because yeast is naturally in our bodies, but it doesn't normally grow when it's in competition with other microbes. But if something happens and it has an opportunity, then it will take over. It'll be opportunistic. Another thing that can cause yeast infections would be like pregnancy, hormone imbalances. When your hormones change, it can change pH, and the microbiota or that flora will change. Another example would be toxin production. So streptococci in the mouth produce compounds that inhibit the growth of most gram-positive and gram-negative cocci. But if something changes, then that can change So E. coli in large intestines inhibits the growth of Salmonella and Shigella as long as you have enough of it in that region. So just like in Biology 103, we talk about those symbiotic relationships. You have commensalism, mutualism, and parasitism. We see the same thing between our bodies and the normal flora or these microbes in our bodies. So these relationships could be commensalistic, which would be that, we're not negatively affected, but we also don't benefit from that microbe. We call that commensalism. This one here, where we are harmed and the bacteria benefits, excuse me, we would call that parasitic. And then the ideal scenario is that we benefit from the microbes and the microbes benefit from us. We think about bacteria in our gut, intestines. They help convert what we've eaten into more absorbable and smaller compounds and molecules so that we can dissolve them into our vascular tissue and we can feed all the cells in our bodies. Like, we have to have bacteria in our intestines to properly digest our food and to get the maximum amount of nutrients from what we eat. And of course, the bacteria benefit from that because they get to live in this tunnel where there's always food coming in. So they get food, and then we get more bioabsorptivity from that food that we eat. So you could think of this as like a gradient. So our relationship with the microbiome from mutualistic, commensalism in the middle, to parasitic at the bottom. So we want these relationships to move up on this chart. We want to move towards mutualistic relationships, not towards parasitic. So the progression of disease could be these microorganisms moving towards that parasitic balance. We get out of balance, and then we start to see the disease show itself. So for our respiratory system, since air that we're breathing in, isn't a place for bacteria to grow. There's not nutrients in it. They can't live from just what's in the air. They need to attach to a surface where there is nutrients to live off of. So the air is relatively stable unless someone's coughing and they have pneumonia and it's getting into the air, or soil has billions of bacteria in it. If wind or animals kick up soil, then bacteria is in the air. But that's very temporary. So you shouldn't expect to just be walking outside and you're just breathing in microbes that are going to give you pneumonia. Like, that's not really what's going to happen. So it's generally pretty sterile. So our respiratory tract, when we get to the lower respiratory tract, it also should be, if not sterile, nearly completely sterile. Now our upper respiratory tract, the nose, the sinuses, the middle ear, they have normal flora because it's so much more readily exposed to where that bacteria is coming in from the air. And ideally, the upper respiratory tract catches those microbes and we're able to deal with them before they can get into the lower respiratory tract. So some different respiratory diseases that we'll look at and some different bacteria that can cause some issues. So one of those groups would be our streptococci. Let's see. So this would be, this would be gram-positive cocci. So they're round and they grow in these elongated chains, right? That strep is like a string. And these cause hemolysis, or we say they're hemolytic. And there's different types of hemolysis. So now they grow best on blood agar, and they can break down red blood cells. So we have alpha hemolytic. So these create this kind of green smear on the growth plate. So it's a green area around the colonies. And then beta hemolytic, it's more clear. And then for gamma, we have no hemolysis or no breakdown of the red blood cells. So for this, alpha hemolysis, that's going to be incomplete red blood cell breakdown, and beta hemolysis is going to be complete lysis of the red blood cells. And that's why beta hemolytic creates these clear areas around the colonies. So some specific examples, we have Streptococcus pyogenes. This would be beta hemolytic, so it creates those clear colonies. It completely breaks down those red blood cells. Pyogenic means pus former. And when you look at this, right, this causes strep throat. If you look in the back of their throat, it has these little pus-filled lesions that are produced on the tonsils. So the person's tonsils will be swollen, they'll be inflamed, and they'll have these white pus-filled lesions on top of them. So one way to tell strep throat from just, some kind of viral infection would be the presence of pus-filled lesions. Now, this disease can progress. It can create what's called a rheumatic fever. This is an autoimmune complication of strep throat. So a virus infected Streptococcus pyogenes, generally going to affect younger individuals. It starts out with just body pains, joint pain, arthritis. It can cause subcutaneous nodules on the joints. And worst case scenario is it progresses into heart problems where it causes inflammation, and it can actually the bacteria will attach to the valves, especially if there's any kind of scarring or damage to the valves. They like to build up on it, and it can actually be fatal if it progresses to that point. It can get to the back of the brain, and where some motor function is processed in your brain, and it can cause something called St. Vitus's dance, which we looked at in class, what it does, but it basically makes your arms and legs kind of flail around because you don't have control of those parts of your body. That does happen in children. It's very rare. It can happen in adults too, but it has happened in children as well. And thankfully, it usually goes away in about 3 to 6 months, but it can stick around. So rheumatic fever, here's just some of the signs, right? We talked about joint pain, heart inflammation, these subcutaneous nodules around the joints, erythema marginatum, and that would be ring-like lesions on the skin. And then that Sydenham's chorea, or what we would call St. Vitus's dance, which is where you get those weird jerky muscle movements. And again, here it says it may even last up to a year. So it can be a while before the symptoms finally reside. So then we have Streptococcus pneumoniae, and this is alpha hemolytic. So it's not a complete breakdown of the red blood cells, it's a partial, and it creates these green colonies on the growth auger. It's diplococcus, so these pairs are surrounded by a dense capsule, which makes them resistant to phagocytosis. Commonly going to be found in the nasopharynx, causes laryngitis, sinusitis, bronchitis. And we got 3 forms here. So you got pneumococcal pneumonia, otitis media, which is a middle ear infection, and pneumococcal meningitis. So inflammation of the meninges, the lungs, and the middle ear. So for otitis media, this would be just your classic middle ear infection. Kids get this more than adults, and it has to do with how their eustachian tubes are. So if you look at an adult, the middle ear eustachian tube that drains the middle ear, it slopes downward in adults. And in infants, as their body's still developing, it's actually smaller, it's more narrow, and it's more horizontal. So it doesn't drain as readily as an adult eustachian tube. So what happens is you can get fluid built up, and that's a very good environment for bacteria to grow. So then they get these ear infections. So it's 85% of children before the age of 3 will go to the doctor for an ear infection. Both of my kids have had lots of ear infections. My son had 3, like in the first 7 or 8 months of his life, and they put him on antibiotics when that happens. That's the just standard treatment, and you want to do that because you don't want it to spread any further, and you don't want it to cause any permanent hearing damage. Next up, we got diphtheria. So for Corynebacterium diphtheriae, this would be a gram-positive, non-endospore forming rod. We say it is pleomorphic, so we have these shape variations. Cause an upper respiratory infection, and we call that diphtheria. Symptoms could be a sore throat, fever, neck swelling, It produces an exotoxin. So while it's still alive, it's producing this exotoxin. It can get into the bloodstream where it can damage your heart, your nervous system, and particularly your kidneys. In our bodies, it creates this gray, leathery membrane appearance that you see in this bottom right picture. And that's fibrin, dead tissue, and the bacteria in these huge clumps or patches. And to prevent this, you can get the Tdap vaccine, which would be for protection against tetanus, diphtheria, and pertussis, which I believe is next. So we have Bordetella pertussis next. So this is obligately aerobic coccobacillus. That's what you see there in that picture. It's kind of like coccus shape, kind of like bacillus, sort of in between there. So we call that coccobacillus. Could have a virulent strain, and those have capsules. So that's going to be even worse. And we say it causes whooping cough, and that's what they call pertussis. Another name for it is whooping cough. So the bacterial cells secrete this endotoxin. It destroys the cilia and the trachea. So the mucus, it's hard to get rid of it. So it accumulates. At first, if a child has this, it might just seem like a cold. They just have a cough. But if, 2 weeks go by and the cough has not improved and it's still there and it's getting worse, it could be whooping cough. And they call it that because as it progresses, when they start to cough, it has this whooping sound to it, especially at the end. And it's kind of raspy, and they're not able to get all this mucus out of their lungs. And then again, the Tdap vaccine would be for protection against pertussis as well. So our next infection would be tuberculosis. We call this Mycobacterium tuberculosis. These are obligate aerobic rods. So it looks like a mold when it grows on the surface, particularly of a liquid medium. They have these waxy lipids on their cell walls. So when they have their acid-fast stain, that carbofuscin stain, it won't be decolorized with the acid alcohol because of these waxy lipids that allow it to retain that carbofuscin stain. So the way someone could get tuberculosis would be inhaling the bacterium, and then it grows in their lungs. So that's where it would start. Once it starts to move through their body, it will, even if most of it is phagocytized, the bacterium can live in these tubercles. That's why they call it tuberculosis. They can do this TB skin test, and they actually will do it for people. I remember when I was at JSU, the athletes would always get tested for tuberculosis. Two outcomes of primary infection: 90% do not even become ill, and the bacteria persist in an inactive state for many years. And then if something happens to their bodies, it will reemerge and cause disease. So they call that post-primary infection or reactivation. So it's already happened, but now it's become reactivated. So let's say they're not eating properly, they've gotten older, they have AIDS, some kind of autoimmune disease. Then the bacteria can escape control of this primary infection and can reemerge. So it can cause chronic destruction of the lung tissue. You might see fever, significant weight loss, loss of appetite. They might even cough up blood as it progresses. The diagnosis is difficult because it grows so slowly. So they can take sputum, which is someone just hacking up some mucus, and they can put that on a culture, but it takes about 8 weeks to actually develop. They can do x-rays to look at your lung tissue to see how much damage has occurred. And that would be that sputum test or sputum sample. Just get some mucus coughed up. The bacteria should be growing in it, so they can grow it on a growth plate. It just takes a long time to grow. And then there's some of that disease pathway for tuberculosis. All right, last one is bacterial pneumonia. So bacterial pneumonias, this would just be those bacteria that cause infections of the lungs, or what we call typical pneumonia. And here's just a list of those. You're welcome to pause and look through. But that is it for our bacterial normal flora and respiratory diseases.